Hi, and welcome to Chapter 6. This is Toxicology, and I'm Sarah Vos. So this chapter is going to walk us through the introduction to toxicology. So we want to wonder, what is toxicology? What is this science? It comes from the Greek word which means poison, and toxicology is the study of the adverse effects of chemicals on biological systems. So it's really where chemistry and biology come together and try to explain why chemicals harm people and animals. And there are two different words that you'll see used a lot. Toxin refers to a chemical that's made inside of a cell. Everything else that's not made inside of a cell is a toxicant. It's important to understand that toxicology is a very interdisciplinary field because you're going to pull from so many different fields like systems biology. And toxicology was really sort of discovered, they say, back in the 1500s by Paracelsus, and his most famous quote is that the dose makes the poison. This is true for a lot of chemicals. It doesn't hold true for some of the newer chemicals that are coming out as endocrine disruptors, and this is a theme that's discussed well in the book, and we encourage you to learn more about this. And when you think about toxicology, you can think about toxicology on lots of different levels. So if you start with the top of that arrow, you can think about toxicology on a population. How many people in a population got cancer because they lived next to a chemical factory? And you can go down that arrow to very finer points of toxicology, all the way down to molecular toxicology, where you're really looking at what does this chemical do to a protein or to DNA. The basis for toxicology, and really the basis for toxicologists to understand chemical risk, is the dose-response relationship. And this is really the relationship between the adverse effect, which you see on that y-axis, and the dose, which you see on that x-axis. And you really want to see a linear response there. That's the type of response that's easiest to extrapolate. Some chemicals do not have that type of dose response curve, and that includes the endocrine disruptors. It's one of the reasons that endocrine disruptors are so hard to evaluate in terms of risk to humans. And when we think about all the chemicals that are around in our world, we can think about a few different ways to classify them. First, you can classify them according to their chemical class, and this is really based on their structure. So these are things like heavy metals, alcohols, or solvents. You can also think about them in terms of the exposure source. So where do the chemicals come from? Are they pesticides that are on your food? Are they air pollutants that are emitted from a chemical factory? And then what I find the most useful is to evaluate chemicals based on target organ. And these can be things that target the liver, which are hepatotoxins or toxicants, or things that target the nervous system, which are neurotoxicants. And then you also consider chemicals in grouping them in terms of their biological effect being cancer or non-cancer. Chemicals that disrupt DNA and can cause cancer are called carcinogens or mutagens or genetic toxicants. And you'll see this theme repeated as you learn more about toxicology and risk assessment, that chemicals are divided into non-carcinogens and carcinogens. And this can be really useful when you're trying to understand what is the effect of exposure to several different types of chemicals. If you know that most of them are carcinogens, you can evaluate a cancer risk thinking broadly about the types of chemical classes that people are, or the types of target organs that people are exposed to. And now we're going to have a few slides on toxicokinetics. And toxicokinetics is really the fate of a chemical. So what happens to any of these toxic chemicals as they are in our environment? How do they get into our bodies? How do they move around in our bodies? And how do they leave our bodies? Toxicokinetics will really determine the internal dose of a chemical. And the internal dose is what is in turn going to dictate the toxicity of the chemical in a human or in an animal. So there are several different steps to toxicokinetics. And first we'll talk about the absorption. So it's really important to remember that when a chemical gets inside of your mouth or is swallowed into your stomach, that chemical is still technically outside of your body. The chemical has to cross through a cell membrane to go inside of your body. So the oral route of exposure is one of the ways that chemicals can get inside your body. If they do pass through your intestinal wall to get into your body, then you can be exposed to them. Another route of exposure is inhalation by the lungs. And so when you inhale a chemical, especially one that's volatile, that's a gas, it can pass directly through your cell membrane in your lung to get into your blood. And you also have another route of exposure, which is dermal exposure through the skin. The skin is a pretty good barrier. A lot of things don't cross the skin very easily. The chemicals that do are often very highly soluble, fat-soluble chemicals that can really dictate how well chemicals are absorbed in your body. And when we think about distribution, we're wondering, where do these chemicals go once they get into your body? And the distribution really depends on the chemical properties and the route of exposure. So chemical properties are very important, because if a chemical is very fat soluble, it might go directly to your fat, and it might bioaccumulate there in your fat. The route of exposure is also important because the oral route of exposure leads to the blood containing that chemical being put directly to the liver. And the liver is the organ that takes care of metabolism, which is the next slide we'll talk about. So when a chemical is absorbed orally, it goes to the liver first. The liver can also often detoxify or get rid of that chemical before it's included in the blood supply to the rest of the body. But if you're exposed via inhalation, when the chemical passes into your blood from your lungs, that blood is then circulated all around your body before it goes to the liver. So sometimes the same chemical by oral versus inhalation can have very different toxicity profiles. And when we think about metabolism, we usually think about metabolism occurring highly in the liver. A lot of other cells can have the capacity to metabolize chemicals, but it does happen most often in the liver. And that's because of a group of enzymes which are called the cytochrome P450s. Those are abbreviated as CYPs or CYPs. And the whole point of biotransformation is to make a chemical very water soluble, because there are two ways that chemicals leave your body, which is excretion. One is through the urine, one's through the feces. So the easiest and the most common route is to make a chemical very water soluble so that it can easily pass into the urine. To make a chemical very water soluble, there are two phases of metabolism. There are phase 1 reactions and phase 2 reactions. Phase one reactions are often meant to make a chemical more reactive. So it's often oxidation, reduction, or hydrolysis, something that adds a functional group. And phase 2 reaction is conjugation. That's where a sugar or some other larger molecule can be added to make the chemical more water soluble. There are a lot of different polymorphisms in people that will make my metabolism different from everyone else's, because we all express different varieties or different flavors of these CYP P450 enzymes. So when we look at a couple of examples of phase 1 and phase 2 reactions, this is where we're really getting into a lot of the chemistry behind or that goes into toxicology. So you see here the structure of acetaminophen, which is Tylenol. You see that in black at the top. You see a couple of phase 2 reactions, including glucuronidation and sulfation. Those are both really great ways to make Tylenol very water soluble. These are excreted right away. And then you also see hydroxylation, and this is due to this cytochrome P450. This phase 1 reaction makes a very reactive intermediate, which you see here in red. This is then conjugated to glutathione. Glutathione is probably a very famous antioxidant. It's made of 3 amino acids. And glutathione, when it's reduced, has the capacity to really pull out a lot of reactive oxygen species, and it has a reputation for that. But there are certain activities that can decrease your glutathione content, such as alcohol consumption. So if your glutathione content is depleted here, and you take a drug such as Tylenol that has this phase 1 metabolism, you really have the risk of building up these phase 1 reactive intermediates, which can be very toxic. In this case, it's toxic to the liver. So this is why doctors tell you, not to take Tylenol when you've been drinking alcohol. So again, excretion is how the chemical leaves the body. The most common is through the kidneys by urine. Chemicals can also be excreted into the liver or bile. If a chemical is volatile and it turns into a gas, then the vapors can be exhaled by the lungs, even if the chemical was absorbed by a different route of exposure. One of the most interesting things about excretion is for fat-soluble chemicals, these can be excreted primarily through breast milk. And this does pose a unique risk for infants. So if you were to look at the chemical content of breast milk, looking at the flame retardants, the PCBs, the perfluorinated chemicals, and conduct a risk assessment on that, it would be a very concerning pathway for exposure. And a lot of infants get a lot of their body burden of these chemicals through breast milk. The last slide here is just about toxicity testing and regulatory toxicology. So with all these chemicals around, we have to have a system to keep us safe from these chemicals. And so this, the last part of the chapter explains the regulatory process and summarizes how scientists understand which chemicals are the most toxic. The first concept that's presented is the LD50, which is a measure of acute toxicity. This is when you give a set of animals several different doses of a chemical and you measure the amount of chemical that kills half of your animals. It is kind of a crude way to evaluate a chemical, but it is very unambiguous. This isn't done very often for the routine chemicals. A lot of this data already exists. More often, scientists are evaluating the chronic or the long-term toxicity. So thinking about developmental neurotoxicity, hepatotoxicity, or reproductive toxicity, and looking at chronic exposures versus acute exposures. You do have the NOEL, which is derived from the dose-response curve. This goes back to toxicology being very important for regulatory decision-making. EPA and other agencies use the NOELs from dose-response curves to calculate the appropriate exposure for humans. And we'll get into this more in some of the later lectures. We do have laws in the U.S. to regulate exposures. TSCA is something that's been in the national news lately. It's right now undergoing reform. It's the first reform since 1976, so it's a very outdated law. Some states have stricter laws, often modeled on the EU regulations, and Vermont is one of the states that has some much stricter laws in terms of flame retardants, lead, and toxic chemicals in children's products. So that's it for now, and have a good day.